Today we will discuss about this STING and BH3 mimetics targeting BCL-XL that trigger apoptosis in this ENKT cell lines. Or what is ENKT cells is extranodal NK cells or T cells lymphoma. So these are the cancer cells that express this high amount of this STING proteins. So these are one of the most probable candidate for this combined therapy that has this STING component and BH3 mimetics. So they first check the STING components in these cell lines, that is NKCL cell lines, NK6 cell lines, or these T cell lines, and along with that they also check is in MEC04 cell lines, and they are able to find out that the STING is expressed in all of these cell lines, and together they are known as ENKT lymphoma cell lines. It is extranodal NK cells or T cells lymphoma. Thereafter, what they did is they knocked down this MEC4 and use the STING agonist to check whether it will activate STING pathways or not. So in order to check that, they check some of the factors of the activations of the STING pathways, such as phosphorylated TBK1, phosphorylated IRF3, and they're able to see these factors in the SNK6, SNT15 and MEC04 that can be seen here in the Western blot for both PTBK1 and PIRF3. But for the STING knockout, they do not see the presence of these phosphorylated pathways. That means the STING knockout does not express this STING signaling pathways because this has been knocked out. This is what they have expected. Thereafter, they check the proliferations of these cells using this different reagent to activate STING signaling pathways in this SNK cell lines. And what they have find out is that in the case of this proliferations, they are able to see a decrease in the proliferations of the cells, but they do not have too much impact on the survivals. And this is the proliferations get affected for all the three reagents that are used for this activation of STING pathways. So indicates that the STING pathways has a role towards this reductions in the proliferations, or has some anti-proliferative roles. And when they use the combined therapy, they are able to see decrease in the cell death also. Combined therapy means they target this BCL-X1 in these ENKT cell lines, because BCL-X1 in the literature has been shown to support the survivals of these cancer cell lines or these lymphomas. So they target this BH3 mimetics to this BCL-XL, and at the same time they trigger, they also activate these STING pathways, and they are able to see decrease in the live cells for all the three reagents. And this result they have observed for SNK6, SNT15 and MEC04. And when they check the time dependent pathways, the time dependent effect on the live cells or different conditions, that is when the this STING agonist is used alone, or BH3 mimetics is used alone, BH3 mimetics is stood alone, or STING agonist is used alone, or the combined therapies are used, and compared it with DMSO. What they have find out is that when they use the combined therapies, the percentage of live cells goes down significantly in all the three cases compared to the individual treatment or the control. This indicates that combined therapies has a more prominent impact compared to the individual therapies. And thereafter they did the experiment on the mice which are injected with this STING agonist targeting BCL-XL, and at the same time there is different reagent for BH3 mimetics. Either individually these BH3 mimetics or STING pathways are triggered, or together, or both of them triggers simultaneously. And when they check the tumor weight, they are able to see that in compared to the case where both the reagents, that is diazoles. So in this in-vivo experiments, they use only one reagent to trigger STING signaling pathways, that is diazole, because diazoles has been clinically approved. So when they use diazoles plus STING pathways, they see a high reductions in the tumor weight compared to the case where they only use these BH3 mimetics targeting BCL-XL. And when they check the impact of the tumors, they are able to see that compared to the individual targeting to the BXL-XL, the combined therapies has a minimum tumor weight compared to the case where individual BH3 mimetics targeting BCL-XL is therapies is used. So this results indicates that these combined therapies of STING and BH3 mimetics has a greater impact or has a higher efficiency of treatment to the tumor cells that can express STING proteins. And in the previous experiments we have understood one more. In the previous experiments or in the previous video we have seen another examples or one more examples of the in vivo. So both of these examples shows that these combined therapies has a better impact, a better effect on the tumor suppressions compared to the individual monotherapies. So that's what they wanted to convey from their experimental results. So I hope you understood the beneficial impact of the STING and BH3 mimetics combined therapy, and what are the kind of tumor cells that are required, what are the kind of tumor cells where these combined therapies has better impact. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.